Hi everyone. In this video we'll be learning about how to make a stained slide. So in biology we use stains so that you can view organelles a lot better. A stain is basically just a coloured dye. The one we'll be using today is called methylene blue. And so when you add the stain to the specimen, it gets into the cells, it gets absorbed in, and it colours the organelles so they're a lot more clear to see. Alright, here's our onion specimen we'll be using. Onions, if we looked at these cells normally without the stain, they'd almost be invisible. We could see the outline of the cell walls, but not much else. So we're going to stain these onion cells today. And onions are fantastic because in between each of their layers, they have this really thin skin. So I'm peeling off one of the layers. On the outside of this layer here, it's really shiny, and that's the skin that we're trying to expose. So I've just pulled off a piece of skin there. You can see that thin layer on the end there. That's the skin we're looking for. And in onions, this layer is only about 1 to 2 cells thick. So it's fantastic for looking at under the microscope. All right. That's our specimen. I've actually got a thicker layer as well, so I'm going to pull off just the thin skin from the end. Okay. Now we're going to put our specimen onto a slide again. I've got a concave slide, which are useful when you're dealing with most specimens because they've got a little indentation in them for the specimen to fit. Okay, there it is. I've already actually got some blue pigment on there from my tweezers, but we're going to stain it properly now as well. So I'm always working over newspaper. I've got butcher's paper down here with newspaper underneath because this stain will stain everything, including the bench. So we put 1 drop of stain on there. That's all we need. I'm going to leave it down there for 30 seconds and wait for that dye to move and infuse into the cell through the cell membrane. All right, we've waited 30 seconds. So the next step is we need to get rid of all the excess dye because if you look at that specimen now, all you'll see is all the blue stain around it. So we're going to get some paper towel. We're going to try and absorb as much of that stain as we can from around the outside. Being careful to avoid getting it on my hands. These stains, they stain everything, including your skin. So we still have a bit of excess stain there. Alright, so I've pretty much soaked up all that excess. And now we have just our specimen left. And I'm spreading it out a little bit. There we go. And then from here, all we need to do is put a drop of water on. A couple of drops of water. And you can see there's actually still a bit of excess stain there. So I'm going to repeat the process, soak up all that liquid again, because we're trying to get rid of as much stain as possible that's not actually inside the cells and stain in the cells. All right, let's put 2 drops on. I'll go for 3 because it's a concave slide. There's a bit of excess area to fill up. We'll take our cover slip. Here it is. Hold it at 45 degrees and dropping it down. There we go. And as usual for a wet mount, there's a little bit of excess water around the edges, so I'll soak up the excess. Straighten the cover slip. And there we go. That's our wet mount with a stain ready to view those onion cells.